So, this is a video for the Biology 1 Enzyme Lab. And the Enzyme Lab, what we normally do in that lab is take a potato, blend the potato up, make say a potato smoothie, because we want to get what's called catechol oxidase. Right? Catechol oxidase is an enzyme. It ends in ASE. Okay, it's found in not just it's found in a lot of vegetables that have starch. Right? Potatoes will turn brown because of catechol oxidase. You know, what catechol oxidase does is it generates a reaction. It converts catechol to benzoquinone, or BQ is another, just a shorter way of remembering that. Right, this is the substrate of the enzyme, of this enzyme, okay? So, we, right, the source of the enzyme is a potato, and what we want to do is see what happens. What's the effect of temperature, or what's the effect of pH has on the activity of catechol oxidase, right? The activity of the enzyme refers to the ability of the enzyme to convert substrate to product, okay? So, right, if we take a reaction like A plus B, right? The substrates are found on this side of the equation, and they become product, right, which are found on the other side of the equation, on the right-hand side. So a chemical reaction, if it requires an enzyme in biological systems, enzymes are required to make this occur, right? You refer to, if the enzyme has activity, right, that it has the ability to produce the product. The only way the enzyme can produce the product is if there was plenty of substrate available, right? Because the substrate is magically converted to the product. Okay? So these are two different chemicals. Now, we're kind of ignoring the oxygen in the water here, right? Oxygen could be thought of as the B. We're not really making AB here. We're converting A to B, to B is probably a better way of saying it, right? This is one molecule, this is a different one. But it doesn't have to always, depending on the enzyme, right? There might be an enzyme that converts AB to A plus B in the reverse direction. Okay, so enzymes are biological catalysts that convert substrates into products. So, in order to measure enzyme activity, right, to measure how much of the product is formed, you could use what's called a spectrophotometer. So that's just a fancy instrument that measures, it gives a numerical value to how brown the solution gets, right? So we'd have this, we take a test tube like this, right? We'd add catechol to it. We'd add catechol oxidase, potato extract to it. And if we sat there and watched it, it'd take it some time. It would gradually get more and more brown over time. If we put it in a spectrophotometer, it would give us a quantity on just how brown it's becoming, or just how much of the product is being produced by catechol oxidase. Now there is a concept called an inhibitor. What an inhibitor does is inhibits enzyme activity. Okay, so if we added catechol oxidase plus an inhibitor, we put all three of them together, we would have very little activity. The inhibitor actually, what happens is, right, the substrate, the way this enzymes work is substrate needs to bind to the active site of the enzyme, right? All right, so I just drew, a little cartoon. This is the protein, and in all of these proteins, there's like a spot where the substrate binds to the enzyme. That has to happen first. And then, right, the enzyme converts a catechol using oxygen, converts it to benzoquinone. It's a product. It actually has a different shape. So the product gets released. But if you have an inhibitor, it can block the active site. Right? This gets in the way and blocks the substrate from reaching the active site. If the substrate can't get to the active site, then the enzyme can't make product. Right? It's that simple, really. Right? It's not that complicated. So inhibitors tend to have like this square part and this square part are similar enough, right, so that this can mimic the shape of the substrate. Now, to get an idea of what we mean by absorbance, if we were to plot absorbance, so just want to briefly explain what this refers to. On this 0 time point, 30 seconds, 60 seconds, 90 seconds, 120 seconds, 50, 150, 180, we would take the absorbance of the test tube. Right? We add the substrate, we add the enzyme to this, it's going to start turning brown. So at 30 seconds, it would be light brown. At 60 seconds, it would start to get darker brown. 90 seconds, it's getting browner, it's going up. Then from 150 to 180 seconds, right, the brown color doesn't change much. But we are measuring absorbance. This is direct. What that means is we are making the product BQ, or benzoquinone. Right? The darker brown this gets, the more product's made. So we're showing enzyme activity increasing for a 3-minute period. That's what this graph is showing. Okay, the absorbance is just a number given, a numerical value given to how much color is produced, and that tells us how much product is produced, right? The more color, the more product, the higher the enzyme activity. Right? That's the job, right? What enzymes are meant to do, their purpose, right, is to produce product. Okay, that's their whole. So every time we need a chemical reaction to occur in a living system, there needs to be an appropriate enzyme to carry that out. So when we talk about glycolysis, it's a biochemical pathway. One of the purposes of glycolysis is to make ATP, and it takes 10 enzymes, and enzyme number 1 makes B, enzyme number 2 converts B to C, enzyme number 3 converts C to D, and so on. Now, we'll talk about that. So this is, now the other part, factors affecting enzyme activity. So, of course, inhibitors, I mentioned already, right? They do exactly what they say. They inhibit the activity of the enzyme. Okay, so that means that reduce the enzyme activity, right? And what we would do in the lab, if we were doing this in lab, is we would put the enzyme in different pHs and ask the question, what happens when the pH is real low? Does the enzyme activity go up or down? If it's real high, what happens to the enzyme activity? Right? We would expect most enzymes to work best in living systems at around neutral pH, pH 7. When they get too low, like pH of 3, that's a very low pH, we would expect that the enzyme would stop making product, maybe, right? Because what happens, if you don't have the right pH or temperature, you might affect the shape of the active site, right? So we were saying the active site is square, but let's say we change the pH, and now instead of a square shape, you get this kind of a shape, right? It's altered, right? That prevents or reduces how much of the substrate, right, gets into the active site. Just by changing the pH. Or even the temperature. We would expect in human beings, right, our body temperature, what is like 100 or 98 degrees Fahrenheit. All right, that equals 37 degrees centigrade. Most of the enzymes in our body work best at that temperature. Now we're using potato extract to get our catechol oxidase, right? And a potato, you know, they live outside, if they're, yeah. So they have to, their enzymes need to function at all sorts of different temperatures. So when we do this different temperatures in lab, it doesn't really start to affect the activity until you get to, you know, above 50 degrees centigrade, which is pretty hot. So pH and temperature affect the shape of protein. That means it becomes, say, more denatured. Now, there is, I'll do another video for, right, there's even a link to it here, lab report video. If you click on that, there's a video on how to complete the what I'll do is I'll give you this data and I want you to make your own Excel sheet graph. So this is, just to explain what this is real quick. If you were to do this in lab, you would add prepare the, you know, add the potato extract with the enzyme and the substrate. It would start turning brown. You would get a value for the absorbance, say it starts at 0.2, at 30 seconds you'd put it in the machine. It goes up to 0.5, it's getting browner, 0.7, 0.1, 0.2. After 3 minutes, the absorbance got up to 1.5. That's at this temperature. Then we do repeat the same thing, except we would expose the enzyme to 40 degrees centigrade first. And that probably wouldn't change the values too much. You know, you'd still get quite a bit of activity. But if we put the enzyme at 80 degrees centigrade, now the enzyme is no longer able to make product, or if it does, it makes very little. Right? You've denatured the enzyme, so the value only gets up to 0.6, right? Which is 3 times less than 0.18 or 1.8. So the temperature, at this temperature, right, the enzyme activity was reduced quite a bit. Why was it reduced? Because of, you would say the temperature denatured the protein, which means the protein starts to, you know, the shape of the protein really changes. So instead of the square like this, now maybe there's no way at all for the substrate to get into the active site, right? And if you can't get the substrate into the active site, right, it's no longer able to make product. Okay, so that's the enzyme lab. And so that's for this week. Be sure you watch the next video for next week. I don't know if it's on diffusion or is it the cell? Maybe it's the cell. We'll talk about the cell. Okay, so all right, keep watching the videos.